So in my lab, we are specifically interested in the question of cancer metastasis, which means how cancer cells spread from the site of origin, for example, breast cancer, to distant organs, for example, the bone, to the brain, to the lung, to the liver. This is important because this is how patients die from cancer most of the time. When the patient have a localized disease, usually it's highly treatable with surgery and additional treatments. But when the disease spread to other organs, often it's incurable. And eventually, most of the patients die from metastatic cancer. So what we try to do in the lab is to understand how does metastasis happen? Because even though it's a very dreadful outcome of cancer, it's actually a very hard thing for cancer cells to do. Out of the millions of the tumor cells that are circulating in the patient's blood, only a tiny, tiny fraction of tumor are able to eventually make a metastasis. So understanding how those tiny fraction of cancer cells achieve the most difficult thing they could possibly do, let us know what are the possible ways to prevent this from happening. My lab have been studying cancer metastasis from two different angles in the past. So one is from the angle of the tumor, or the so-called seeds. The other is the angle of the stroma, the tissue environment, or the soil that allow the seeds to grow. And this hypothesis is called seed and soil. Over the last 20 years, since I started my lab in Princeton, the focus is mostly in the seeds because that's easier system to study. The other big question, of course, is the soil. In the past, that has been mostly being a black box because to understand soil, we have to know what's in the soil. This is a key question. What's the composition and the property of the soil that allow metastasis to happen? But we don't really have the tool to study that until very recently. And that's the new direction my lab is getting into now. So what we are doing now is using a lot of the state-of-the-art single-cell spatial genomics, spatial transcriptomic analysis, and then we are working with Josh in a lot of ways, Princeton branch to integrate the spatial metabolomics, so that we will have really a three-dimensional view. And actually in the future, it will be four-dimensional view with the time as another dimension to see how the soil evolve and not only understand what's the composition of the soil, the cell types exist in that, what's the molecular signaling that's going on, also the metabolites that is being used or generated in this process. That I think is going to give a lot of the new exciting, potential therapeutic targets that we can intervene. Well, being part of the Ludwig global family really give us access to many of the resources, collaborations, expertise that would not be expected to have at Princeton. I actually visited the Oxford branch last year. After many conversations, there are really several directions for us to collaborate with different branches like Oxford, Lausanne, and also a lot of centers in several different areas that are close to our heart, but also we can really take advantage of the expertise. Our ultimate goal is to translate our basic science discovery in the laboratory into medicine for patients. I think that's the ultimate test and proof of the rigor of the science that we are performing. The discovery we make in the lab eventually will make into new medicine, and that will either target those previously untreatable disease that are already stage 4, or turn some of the less effective therapies more effective for cancer patients.